So both evolutionary biologists and ecologists have long been fascinated by the diversity of secondary metabolites in plants. And a lot of this interest started off with Dojansky coming up with some ideas thinking about how species interactions could actually be associated with diversity, latitudinal diversity gradients in species. Later on, this idea was formalized into the latitudinal herbivory defense hypothesis. This hypothesis is a fairly well-known and controversial one that suggests that plants at higher elevations, or at lower elevations, should have greater levels of defense. And the basic reasons for this is that at lower latitudes, we find greater species richness, greater species abundance, and probably greater species interactions. And those species interactions can create an increase in herbivory and selection for greater plant defense. If there's also a cost to these different defense compounds, then at higher latitudes, where there may not be as much herbivory pressure, you might see less defense evolving. You might therefore have a latitudinal cline in these plant defenses, getting some pattern that looks like this, where the red is greater levels of defense in plants. This was really articulated well and tested first by Don Levin in 1976, who looked at a bunch of different plant communities all over the world and found this pattern was fairly strong. We looked at the number of plants, or the percentage of plants that contain uploads. This kind of hypothesis didn't have a lot of empirical testing done until the last 10 years, when it exploded. And one of the reasons it exploded is because Angelina Moles did this really nice, a nice meta-analysis, where she looked at plant communities all over the globe again, and looked at several general defenses in these plants, and found real no associations that showed evidence for the latitudinal herbivory defense hypothesis. And of course, everyone coming out of the woodwork afterwards trying to explain why she didn't find anything here to bring back kind of this hypothesis. And there's a lot of reasons why you wouldn't actually find anything surveying the way she did. And there's several challenges to this, like for instance, it's hard to find individual widespread species that are present over a latitudinal range that is large enough to actually see this pattern. There's many different kinds of defenses. There's introduced versus constituent defenses. There's generalist and specialist herbivores. And all these things suggest that there may be different patterns over a latitudinal gradient. And finally, in the 90s, there was a large debate among plant herbivore people that shook their fists and said, we don't even know if there are costs to these defenses. We haven't been able to prove this. But when I think about the latitudinal herbivory defense hypothesis, I'm not thinking about this pattern necessarily all the time. I'm thinking about, this is a question of adaptation, and thinking about ecological trade-offs. A trade-off for defense versus resource allocation to growth and reproduction. And I was thinking about studying this in another widespread species, my mimulus species. So what I'm going to talk about today in this widespread species is whether we see this pattern, this latitudinal cline in chemical defenses across latitudinal and elevational gradients. And I'm also going to talk about a couple about the associated underlying ecological trade-offs that are associated with either defense or growth and reproduction, using both the common garden approach and phenotypic selection analysis. So the species I'm using for this is the ecological genetic model system, Mimulus guttatus. And it's an ecological genetic model system because it has a number of sequence genomes, molecular toolbox, all of these different great factors for using it for those kind of studies. But in our case, what really we care about is that it has a large series of both chemical and physical defenses, and it has a very wide geographic range spanning a large latitude, degree of latitude. So the major chemical defense in monkeyflowers is these phenylpropanoid glycosides, or PPGs. The two main ones are chilandroside and calceroside A. And they both, all these PPGs span off of this phenylpropanoid pathway. It's the first basal branch of that pathway. And unlike many plant defenses, this actually makes up a large component of dry leaf weight. On average, if we look throughout all the populations that I've ever surveyed, it's about 8% of dry leaf weight, which is fairly high for a defense and secondary metabolite. The other nice part about the guttatus system is I sample across regions that have substantial variation in growing season length and time throughout the range, because the annual monkey flowers that I look at require water essentially to be growing. And they live in these Mediterranean climates. So growing seasons start with snowmelt or spring rains, and the growing season ends with these terminal droughts. So there's a defined length of growing season here, and if you look throughout the range, it differs widely from about 6 weeks up to about 4 months. And they look like this sometime in August, nearly all the populations do. And the nice part about having this substantial difference in growing season length is that you could predict that the cost of defense would be associated with it. In environments that have really short growing season lengths, we would predict that you wouldn't want to allocate a lot of resources toward defense because you'd be trying to grow and reproduce as fast as possible to finish your lifespan. So we did in Utatis, we did a common garden experiment where we went out first and sampled along 35 populations along a 14-degree latitudinal range from Southern California up through Central Oregon. We brought all these seeds back. We also sampled along 2 elevation gradients, one in the Sierras in that rectangular box, and one in the Cascades in that other rectangular box up there. We brought all these seeds back to the greenhouse and grew them in this common garden experiment. We had constitutive treatment where we didn't do anything to the plants, and we had a damage treatment where we did a paper hole punch to simulate herbivory and think about induced defenses. At a certain developmental stage, we then flash froze several different leaves for HPLC analysis downstream. So what did we find in this? So starting off, that damage treatment I just told you about didn't actually do anything in these annual plants, which isn't unexpected because these are annual plants that don't have very long periods of very long growing seasons. So we see that actually damaging the plants via paper hole punch actually even decreased the levels of some of those compounds. But more towards the main period question, we found that there is extreme variability in both the abundance and arsenal of these PPGs across the latitudinal range that we sampled. You can see this really easily, even when we put it in pie charts that are the size is dependent on how many or average level of PPGs in the population. And you can just see in the difference in colors, where you see more of those orange in the pie charts towards the southern edge of the range, and more blue towards the northern edge of that range. But to formally compare this, we're remembering that as expectation for the latitudinal herbivory defense hypothesis is that we have greater levels of defense in lower latitude populations. We find a number of PPGs that oppose this hypothesis, where we find greater levels of defense in high latitude populations relative to low latitude populations. But we find mixed results. We find other PPGs that support this hypothesis, where there's high levels of defense in low latitudes relative to high latitude populations. And finally, we find other ones that have no associated pattern with latitude or elevation, but they are at extreme levels in some populations versus others, which is really interesting. If you look at the overall level of defense in this, it actually shows a pattern that opposes that latitudinal herbivory defense hypothesis, where there's greater defense in those high latitude populations. And just to give you kind of a summary right here, most of the PPGs that we surveyed oppose this, a few supported it, and that Verbascus side had that crazy pattern where there's a couple populations with 30 times the level. So let's think about the ecological trade-offs that actually could cause these patterns that we see here. They could still be the same trade-offs that cause patterns along the latitudinal gradient. So one of those could be herbivore pressure. So I surveyed a bunch of populations along that latitudinal gradient to see what the level of herbivory was, leaf herbivory, in each of those populations. Controlling for development, we do find that higher latitude populations have greater herbivore pressure than lower latitude populations. And this kind of makes sense when you think about the climates of Oregon versus Southern California and where these plants actually live at. And this could be an explanation for that climate. But I also told you that I surveyed a bunch of populations over different, in areas that have different lengths of growing seasons. And there could be that cost of defense that could also generate these clines. And we also see evidence for that, where there's greater levels of PPGs in areas that have longer growing seasons as well, implying that there is this cost here. You can actually compare this within traits too, to look at correlations between traits. And for instance, if you compare the levels of total PPGs to time of flowering in these populations, we see that plants that have great defense also tend to flower later as well, suggesting there may be some sort of genetic correlation between these two traits. So, I told you now about the kind of patterns that we had across the landscape, but we're kind of unsatisfied at this point with ecological trade-offs because we're just serving a bunch of populations in a common garden. This isn't natural populations. And we really like to know something about actual fitness in this case. So we brought it out to the field. So I had a couple extra questions in here too, and this is basically, we want to quantify these trade-offs. We want to understand if there's selection from greater defense or plants have higher levels of defense. And we want to understand how genetic correlations actually facilitate or constrain adaptation in the system. So we brought this out to the field, and we did a series of reciprocal transplant experiments at high and low elevation in Oregon, where we had the highest levels of herbivore pressure and defense. Each one of these populations, we planted several lines from 4 parental populations a low elevation population from Oregon, a high elevation population from Oregon, a low elevation population from California, and a high elevation population from California. And we also did we crossed these to make mapping populations. So we crossed the low and high elevation from Oregon and the low and high elevation from California and bred these into outbred F4 lines to do mapping these populations. Now, just to give you an idea of what the defenses look like in those populations, they're dramatically different in abundance and arsenal between these different areas. And in each one of these reciprocal transplant sites, we measured about 25 traits. This, I'm not gonna lie, this experiment was not originally done to look at this growth defense trade-off. But we measured, including those traits, we measured herbivory at flowering, we measured growth rates and plant height, we measured flowering time, and we measured a bunch of different fitness components in each of these two populations. And we had these expectations that potentially, because we know that California, the low elevation plants and high elevation plants from California produce less defenses than the plants from Oregon, we would expect them to have the plants from Oregon to have lower levels of herbivory in the field. Also, you could expect that there might be some kind of local defense is better than native defense is better in those particular regions. So the high elevation plants may have lower levels of herbivory in the high elevation side than the low elevation plants because they have similar levels of defense as well. Right. And you could make a prediction about local adaptation there too, where if herbivory mattered quite a bit and defense mattered quite a bit, we would see the kind of reciprocal patterns here. We look at fitness, number of flowers, that we saw for patterns of herbivory. So what do we actually find? Sorry for the little bit of light points here, but we do find that leaf damage expectations from our common garden experiment were plants that had lower levels of defense in that common garden experiment actually had higher levels of herbivory. But that did not exactly match what we found for patterns of local adaptation in those sites. We actually found at the low elevation site, the low elevation population from California had the highest fitness. At the high elevation site, we found the population from high elevation population from California had the highest fitness there, which is a really interesting pattern given Amy's talk earlier that she gave a meta-analysis on the same kind of topic. And when you bring it down to look at those actual mapping lines, we find even kind of more paradoxical results, where we find this relationship between fitness and herbivory, where plants that had greater herbivory actually had higher fitness in some of for instance, the California cross in the low elevation site, and the Oregon cross in the high elevation site. So this is kind of paradoxical. It's the opposite of what you expect if herbivory was actually driving that kind of relationship. But if you look at the correlations between the different traits, we find that there's a trade-off between plant height and herbivory, or presumably plant height in defense in this case too, where plants that are taller have greater levels of herbivory, plants that flower earlier have greater levels of herbivory in certain sites, and those are really the traits that are driving patterns of local adaptation in this site, where plants that flower earlier are now taller, so have a faster growing rate. Those are really what's driving that difference in fitness between these different populations. So this implies that these correlations between defense, reproductive timing, and defense have large effects on local adaptation of those sites, and defense may or may not be in their selection here. I also should mention this was done in a drought year where the climatic conditions in California or in Oregon actually matched those in California better than what they would from the historical averages in Oregon. So in conclusion here, latitudinal herbivory defense hypothesis, right trade-offs, wrong pattern it seems. There's both these costs of defense that we see from the common garden experiment. There's a strong relationship that we see both in kind of observational studies and in field populations between herbivory and defense. And we see these kind of genetic correlations between growth, reproductive timing, and defense that drive these local adaptations. And we see different ways that these annual monkeyflowers are doing it throughout their range. In different areas, some plants have earlier flowering time, but also high biomass or high defense. And we do see these same trade-offs that extend in this annual perennial division in monkeyflowers too, with these same trade-offs. My future work is actually breaking these down into different defenses and looking kind of at fine-grained local adaptation with these different defenses in herbivore communities and looking at this at a genetic level. With that, I'd like to thank my collaborators, Liza Valesky and Ben Blackman, who helped with this work, and funding from NSF and American Genetics Association. Thank you.